Good afternoon. Wow, that was quick. That never happens. I am so pleased to welcome all of you on this very special occasion for the School of Medicine. While there was a virtual celebration on April 2nd, 2021 during COVID, it is truly a pleasure for us to now formally carry out the installation together in person and to celebrate this wonderful occasion. It is such a privilege for us to recognize the incredible impact of the two people who we are recognizing today, Jeffrey H. Miner as the first to hold this new established professorship, and Dr. Eduardo Slatopolsky, in whose name we will be recognizing this professorship. And I want to welcome Dr. Slatopolsky and his son Daniel and his daughter Andrea. Wonder if we could all give a round of applause for that Slatopolsky family. And it is also a pleasure for us to have Jeffrey Miner's family, including Jeffrey is joined today by his partner, Larry McDonald, his mother, Elaine Miner, his sister, Tricia Miner, his sister and brother-in-law, Dana and Ari Greenberg, his niece and nephew, Sam and Elise Greenberg, and his partner's brother, Keith McDonald, and many, many friends and colleagues who are here today. Let's give a round of applause for the Miner. And we're live streaming today, so we also have many friends and colleagues, and it's so wonderful to see many people who are here, both for Jeff and for Eduardo. This is just a fantastic celebration for us. Dr. Frazier will say more about Dr. Miner in a few moments when she formally introduces him, but I just want to comment from the perspective of the school and the university how thrilled we are to recognize your accomplishments, Jeffrey. Your work on the molecular characterization of the glomerular basement membrane is leading the way for us to understand how the kidney functions and the basis for how we might target better therapeutics for kidney disease. Your work and what you have accomplished is so emblematic of the deep quality and broad impact that characterized Eduardo's career and is so fitting for the inaugural naming of this professorship. Before we bestow the battalion, please allow me to share a few highlights about Dr. Eduardo Slatopolsky, Emeritus Professor of Medicine at Washington University. The professorship honors Dr. Slatopolsky and his wife, Judith, who died in 2012. They were married for 52 years. Dr. Slatopolsky's legacy has been incredibly impactful, and this professorship will continue that into perpetuity. Dr. Slatopolsky graduated from the University of Buenos Aires, Argentina in 1959, and eventually came to our university as a postdoctoral trainee in the renal division in 1963. By 1967, he was the director of the Chrome Alloy American Kidney Center, a position he held for 30 years. He remained active in his research lab until his retirement in 2016. Dr. Slatopolsky dedicated his career to research studies on how kidney disease led to bone loss, and this led him to important discoveries about secondary hyperparathyroidism, hyperphosphatemia, and vitamin D deficiency. This work had far-reaching impact among many of his colleagues and friends who were here today. Indeed, his lab worked with Hector DeLuca in the discovery and development of paracalcitriol, a compound that is now used to prevent secondary hyperparathyroidism in patients with chronic renal disease. I want to mention that the licensing of this technology has brought quite significant funding to the university, and I want to also mention that we've used this funding to provide new support for the Office of Technology Management and for providing more services to our faculty for commercialization, another incredible impact of your career, Eduardo. And so we have much to be deeply grateful for your and your family's investment in Washington University over many, many years. And the School of Medicine appreciates this opportunity to recognize that legacy and the impact your work has had on human disease, your support for our venturing into the future in commercialization, and for this extraordinary philanthropic gift. Our sincere thanks to you for making this day possible. It is a special honor to be with you as we officially install the inaugural Eduardo and Judith Slatopolsky Professor of Medicine and Nephrology. So Eduardo, may I ask you to come forward now, and please join me in recognizing. You're doing fine. Take your time.
Yeah.
Come over here for a second.
Yes.
Yeah.
I want this.
Okay.
Yeah.
Come over here so we can
Sure.
take a picture of you with the medallion. And the medallion reads, "In grateful appreciation presented to the Slatopolsky family, October 27th, 2022. You want to say a few words?
Yes.
Come here.
Dr. Perlmutter, Dr. Frazier, my dear friends and relatives, I'm very happy to be here today to celebrate our friend Jeff Minor. A half a century ago, my wife Judith and I came to St. Louis, and I joined the renal division. But at that time, I didn't know how to work in the lab. I had no experience. But I was very lucky because I have a superb mentor, Dr. Neil Bricker. We realized at that time, although we improved our knowledge in the laboratory, that our patients were dying. There was no dialysis here at Barnes Hospital, in St. Louis or in Missouri. Then Dr. Bricker asked me to go to Seattle and to learn from Dr. Scribner dialysis. I came back, I set up the unit, and the unit was one patient, one nurse, and me. That was the whole unit. Over a period of time, there was a geometrical growth in these parameters. And today, there are more than 115 units of dialysis all over St. Louis. And I asked last night to Dr. 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 Marcos, right, thank you. Thank you, Marcos. Dr. Marcos is my friend. He is always, because I never call him Dr. Ronste. I always call him Marcos. I don't remember the name Marcos. I called Dr. Ronste and said, Marcos, how many patients are we having dialysis in the United States today? And we have close to 800,000 patients. Of course, we didn't take care of these 800,000 patients. Many physicians were trained, and now we are doing, taking care of this very large number of patients, and they're maintained alive. Over the years, with the support of Washington University and my wife of 53 years, I was able to achieve what I always want to be, a scientist. Washington U is unique in the sense that you have collaborators, resources just around the corner. And Washington U gave me the best 50 years of my life, the best 50 years of my life. Then I feel that today I should be able to do something and go back to the university. And, after 50 years of good life, the minimum I can do is to help with this chair. Dr. Frazier, in a few minutes, she will describe in great detail the scientific achievements of Dr. Jeff Miner. And I'm not going to say it here. But Jeff is a genuine person. Jeff is a solid person. He is a gentleman among gentlemen. And for those of you who speak Spanish, he's a mensch. Now, if you don't speak Spanish, you're in trouble. You have to understand Spanish. That I will speak then, and then I will explain to you what do I mean by mensch. We are so happy to have him in our division. Every time I went to his lab and I asked for very difficult questions, very difficult problems, he never said no. He stopped thinking, and he gave me good advice. Jeff is a person who never gets sick, never gets tired. He never gets upset. He never raises his voice. He's a good guy. He's a real good guy. And I'm proud that now my name and his name are going to be together through this endowed chair. Jeff, my friend, my colleague, I'm very happy that you are the one who's going to get this chair. Congratulations.
It's now my pleasure to introduce Dr. Does she need an introduction? To welcome Dr. Victoria Frazier, the Adolphus Busch Professor of Medicine and Chair of the Department of Medicine, to formally introduce Dr. Miner. Vicki.
I'm going to keep my mask on because the last time I was up here and took my mask off, my hearing aid went flying over onto the floor, and that was embarrassing. So this is safer. It's my pleasure to introduce Dr. Jeff Miner, Professor of Medicine and of Cell Biology and Physiology, and now the Eduardo and Judith Slatopolsky Professor of Medicine and Nephrology in the Department of Medicine. Jeff earned his PhD in molecular biology and biochemistry at the California Institute of Technology. He did his postdoctoral training here at WashU in the neurobiology lab of Dr. Joshua Sens. As a postdoc, he investigated the molecular basis of nerve-muscle interactions. His original goal was to analyze the structure and function of the basement membrane that forms the synaptic cleft material at the neuromuscular junction. But really, fortunately for us, he fell in love with the kidney. And due to similarities with the kidney glomerulobasement membrane, Jeff's experiments led him to make major inroads into understanding how basement membranes are involved in kidney development, function, and disease, a topic that has continued to be the focus of his investigations throughout his career. Following his fellowship, we were fortunate to recruit Jeff to join the faculty at WashU. In 1996, he was 12 years old then, And he has become increasingly successful here, becoming professor of medicine in 2007. Jeff has made seminal contributions to our understanding of basement membrane biology in his 26 years as faculty in the Division of Nephrology. He's an international leader in basement membrane biology and how basement membranes are involved in kidney function and disease. He is an expert in research on Alport's syndrome, a genetic disease of the kidney glomerular basement membrane. His lab is focused on understanding why the kidney fails in Alport syndrome and how to slow or stop kidney disease progression. His research combines the use of mouse models of the disease with state-of-the-art genetic and imaging approaches and direct testing of drugs as potential therapeutics. Jeff's research is published in the highest impact journals for nephrology, including Proceedings of the National Academy of Science, Journal of Clinical Investigation, Journal of the American Society of Nephrology, and the American Journal of Physiology. He's published over 190 peer-reviewed publications and has been invited to speak on his research nationally and internationally. Jeff has been continuously funded by the NIH for 26 years. He currently has 2 R01s and R43 and serves as director of the P30 Transgenic Embryonic Stem Cell Core and director of the Precision Animal Core for the Digestive Disease Research Center. In 2019, he was awarded a large core initiative by the Children's Discovery Institute to fund the Pediatric Disease Mouse Models Core, which generates mouse models for pediatric disease research using state-of-the-art CRISPR-Cas9 technologies. Jeff is a really sought-after mentor, having served on 16 qualifying exam committees and 31 thesis committees. 2 of his postdoc fellows, Maggie Chen and Hani Suleiman, are now NIH-funded physician scientists and faculty in the division. He serves on the ASN Career Advancement Committee and has served on numerous NIH study sections, including as chair of the NIDDK Fellowship Review Grants. He also reviews for the United Kingdom Research and Innovation Medical Research Council. Jeff is active in administration and leadership roles, both locally and nationally. He has served as Director of Basic Research in the Division of Nephrology since 2016. And in that role, he redesigned and formalized our mentorship guidelines for trainees. He also co-directs the Division of Nephrology's Renal Disease Training Grant, a very important mechanism for training young scholars. He serves as the chair of the Medical Schools Committee on Fellowship and Awards and has served as vice chair and chair of the Basement Membranes Gordon Research Conference and currently serves as president of the American Society for Matrix Biology. On a personal note, Jeff is a loving partner to Larry McDonnell and a very caring and supportive son, brother, and uncle to his wonderful extended family. Jeff is a beloved member of the renal division, as you heard from Eduardo Slatopolsky. He is incredibly steadfast, kind, compassionate, caring, and always puts the needs and the welfare of others before himself. He's a wonderful mentor and teaches research ethics and has been very engaged in promoting equity and inclusion in the division, the department, and the School of Medicine. Please join me in welcoming Dr. Jeff Miner, who will present Can a Damaged Kidney Filter Be Fixed? The Paradigm of Alport Syndrome. So before Jeff's talk, before Jeff's talk, Dr. Perlmutter will bestow the medallion.
Come over here. We got a little ahead of ourselves here. So this is the medallion that signifies your appointment, Jeffrey, and it says on the front, Eduardo and Judith Slatopolsky, Professor of Medicine in Nephrology, Washington University in St. Louis, and on the back, Jeffrey H. Miner, PhD, FASN, October 27, 2022, and And here you are.
I get to give you a copy.
Are we on schedule? Dean Perlmutter, Dr. Frazier, Dr. Humphreys, Dr. Slatopolsky, and family, distinguished colleagues, family and friends, here and on the live stream. I'm so appreciative and humbled by this great honor. I thank Eduardo and his family and Washington University School of Medicine for bestowing it upon me. I regret that I never got to know Judith, but I know what a great light she was for her family and for her community. I first met Eduardo during my nephrology division job interview in early 1996, after which I learned that he was, and still is, a legend in terms of what is known about how kidney disease causes mineral and bone disorders. During our 30-minute interview, Eduardo tried to explain to me everything you see on this slide. As he was explaining it to me, I remember thinking something that I hope I didn't say. I thought the kidney was just a filter. I'm going to begin my presentation by thanking those without whom I would not be here today. I think I said that right. My parents, who gave me the inclination to go to college from a very early age. They came to my graduation, of course, at Northwestern University in 1985, and to my graduation with my PhD at Caltech, along with my grandmother, Pearl. And it was exciting, especially exciting because President George H.W. Bush was the commencement speaker at that time. Though, as you can see, even then I was leaning a little bit to the left. So I wouldn't be here today without my amazing, brilliant PhD thesis mentor, Barbara Wold, at Caltech, who taught me many things. Barbara's office was basically the lounge for all the graduate students in the lab. There was nowhere else to go, and we had hours and hours of conversations about science and other things. But one thing that I've always remembered Barbara talked about or taught me was to focus on the questions and carry out the experiments that will reveal the way to the truth and the light. That was kind of her own buzz phrase. And so I've tried to do that throughout my research career. I came to Washington University in 1992 to do a postdoctoral fellowship with Joshua Saines, who has since left and moved to Harvard. But Josh is a brilliant neuroscientist who let me work on kidney disease in his lab, which not every brilliant neuroscientist would do. But that's where the science took me, and I think he saw the value in that. Josh taught me that great antibodies and images, plus the right gene-edited mice, can help me find the way to the truth and the light. In the fall of 1995, Josh came to me and said, "Jeff, I was invited to give a talk in nephrology, but why don't you do it? If you give a good talk, they'll offer you a job." A few months later, Mark Hammerman, then chief of the renal division, called me and asked me to interview for a position in the renal division. And I, of course, accepted, and the rest is why I'm here today. And it was a risk, I think, for Mark, as well as for me, because I was a PhD. There were no PhD tenure track professors in the renal division at the time. So I was the first of several others that had been hired. And I think I'm glad Mark took that risk. So I wouldn't be here today without all the people that have been doing research in my lab over the last 26 years. This is actually 4 years into my having a lab, and this was the group then. It grew and shrank and grew and shrank over the years, and here's just some snapshots of people that I hope some of them are in the audience or watching on the live stream. It's been such a pleasure having so many hardworking, smart people who know more than I do about certain things and are willing to learn new things from me. This is the final picture taken last year during a COVID lunch outside in my condo complex. And we're all wearing octopus t-shirts for reasons that will become apparent quite soon. And here's a list of all the people that have come through my lab, graduate students and postdocs. The ones highlighted in red are now on the faculty at Washington University. So I thank all of them. I also thank incredible support staff, as well as the staff that has helped me make transgenic mice over the years, Mia Wallace in the Mouse Genetics Core, as well as Renata Lewis, who has since retired from WashU. I wouldn't be here today without Andre Shaw. He's a brilliant immunologist who lucked out when his knockout mouse showed an unexpected kidney disease phenotype. The same thing kind of happened to me. I still remember Mark Hammerman calling me into his office in late 1998 so I could meet Andre and try to interpret his electron micrographs. This led to the start of a very rewarding collaboration, including my first paper in Science, one of the top journals in all of research, a set of other impactful publications that followed, and a wonderful friendship with Andre and his wife, Cindy. When Andre left WashU for Genentech in late 2015, he transferred his R01 grant in its 16th year to me to continue as the PI. It was an incredible gift that kept on giving, and I can't thank him enough for that. And there's the grant number. I wouldn't be here today without wonderful collaborators and career supporters. Aubrey Morrison, Robert Sr., Helen Liapis were instrumental in teaching me about how the kidney functions, how basement membranes are important in the lung, and how to interpret kidney pathology. Rafi Kopan, who's here, taught me so much about developmental biology, and thanks for being here, Rafi. And others, Sanjay Jain, Jonathan Barnes, Guy Jenin, and Ben Humphreys have been incredible collaborators. There are a number of collaborators in Nashville at Vanderbilt who have been so helpful to me during my career. I'm happy that Amber Pozzi and Roy Zent are here today. And then collaborators from and supporters from around the world who have taught me so much and allowed me to become involved in their own projects. And I wouldn't be here today without your and my generous tax dollars. That helps fund the National Institutes of Health, which has funded the great majority of my research, primarily the NIDDK, but also NIAMS and the National Institute of General Medical Sciences. And then also private foundations that have been terrific supporters of my research endeavors, including the March of Dimes, American Heart Association, the Alport Syndrome Foundation, Kidney Research UK, and the Children's Discovery Institute here at Washington University and St. Louis Children's Hospital. So now I will move on to the science part of my talk. And those of you who are here from Wine Club know what an educator I always try to be, whether it's about wine or about kidneys. So my goal is to make sure that everyone in the room learns something about the research that I do. So here's a short introduction to the kidney and the nephron. So most of us have two kidneys that are located near the back of the abdominal cavity. And this is what a healthy mouse kidney looks like. It's what a mouse kidney looks like. It's not a real kidney. It's not even a picture of a real kidney. It's a picture of a kidney bean that I found online that easily could fool me that it's a mouse kidney. Now, which came first, the kidney bean or the kidney? I'm not quite sure. And here is a cross-section of a real human kidney showing how the red part packed with nephrons that do the filtering and making of the urine, and these cavities that collect the urine, and then the ureter that pumps the urine towards the bladder until we can find the right time and place to get rid of that urine. And here is a diagram of the nephron. So the human kidney is composed of about 1 million of these individual nephrons, which are individual filtration units. The filtration part of the kidney that I study is called the glomerulus, and it's shown here as a very vascularized, it has a very high pressure blood flow system of capillaries that also contains the glomerular filtration barrier between our bloodstream and the urinary space. There are other parts of the nephron that I don't study called the tubules that the primary filtrate from the glomerulus enters through here, and the cells that line that tubular portion of the kidney resorb molecules, salts, proteins, and water that we don't want our body to eliminate. So it's kind of a retrieval system that's very important. So here's a more schematic view of the glomerulus. A cross-section view, you're seeing cross-sections of the glomerular capillaries here. So blood would be flowing through these capillaries, and then the urinary space across this glomerular filtration barrier is in this region surrounded by an enclosure called Bowman's capsule. There's a specialized cell type I want to talk about today called the podocyte, which sits within that urinary space and sits on top of the very important extracellular matrix structure that I study called the glomerular basement membrane. So those podocytes, they help synthesize that glomerular basement membrane, and they basically sit on it throughout our lifetimes and maintain the function of the glomerular filtration barrier, which again is this three-layered structure here. So I think there's no better way to show how something works than to show a movie of it actually working. And this I will show you soon is a movie of a live rat whose kidney has been exposed out the back of the rat and put under a fluorescent microscope. And that microscope can see pretty deep into the rat kidney, and there are glomeruli near the surface. And this rat was injected with two different tracer molecules. One is large and yellow colored, one is small and red colored. And so I hope the movie plays, and indeed it does. And so this is demonstrating glomerular perm selectivity. The large yellow tracer remains in the glomerular capillaries, whereas the red tracer is very easily filtered from the bloodstream into the urinary space, where it then goes down the tubular portion of the nephron, and these cells lining that portion, the tubule, will eventually change it into the final urine that needs to be excreted. So lately, when I talk about the kidney filter, I make an analogy. I've been talking to patients, Alport syndrome patients, quite a bit about how the kidney works, and so I decided I needed an analogy, so I used the coffee filter. So the coffee pot, the coffee that many of us like to drink, and the coffee filter. So that coffee filter, as I'm sure you all know, has very small pores within it. This is probably a quote-unquote gold filter that has made of metal, and there are tiny pores in that metal, and the job of those pores, of course, is to keep the grinds from flowing into the coffee. And so the coffee filter allows the passage of water and small molecules, such as caffeine, flavor, color, notes of toffee and walnuts. But it also restricts the passage of grinds into the filtrate, that is the coffee. So the kidney glomerulus has somewhat of a similar role, but of course the players are a bit different. The glomerular filtration barrier allows the passage of water and small molecules, such as salts, small proteins, nitrogenous waste products from regular metabolism that become the final urine. So those substances are initially within the bloodstream, but they're able to cross the glomerular filtration barrier into the urinary space. But that barrier is important to restrict the passage of red blood cells, which are these black things, and protein, indicated by the P's, into the filtrate. Now there's always a little bit of P that sneaks through, and the tubules are designed to resorb that. But if there's any sort of defect in the glomerular filtration barrier, lots of P's can sneak through, or lots of proteins can sneak through, and ends up into the P. So I mentioned the podocyte being a critical player in the glomerular filtration barrier. This is an image made by someone I forgot to credit, a very talented German microscopist. And the podocyte is this cell here. It has a cell body, and then it extends processes that are called major processes because they're large, and those major processes then extend other processes that are smaller, and those are called foot processes. So these podocytes are unique octopus-like epithelial cells, and as I mentioned, they make the glomerular basement membrane, which is the blue ribbon-like structure here within the barrier, and they reside on it. Injury or loss of this cell can cause kidney disease that can eventually lead to kidney failure. Here's yet another view by scanning electron microscopy of a podocyte. So this is the main body of the cell, but it extends these major and minor processes called foot processes. If one takes a slice of this and looks at it under an electron microscope, this is what the glomerular filtration barrier looks like. There's blood on one side flowing under high pressure. There's the urinary space on the other side. And then this is the three-layered filtration barrier between them. And I'm going to focus on the glomerular basement membrane. And somewhat on the podocyte above that helps make the majority of it. So, as was mentioned by Dr. Frazier when she introduced me, Alport syndrome is the major disease that I study and we'll talk about briefly. So Alport syndrome is a hereditary glomerular disease, usually accompanied by serious hearing and manageable eye defects. The incidence is estimated at 1 in 2,500 to 5,000. It's often diagnosed in children based on the presence of blood in the urine and typical defects in the GBM, as observed by transmission electron microscopy. It almost always progresses to end-stage kidney disease, or ESKD, by late adolescence or thereafter. Progression is signaled by the onset of proteinuria, meaning those Ps, those proteins that shouldn't leak into the urinary space do so, and that causes problems for systemic problems within the body. But treatment with drugs that lower systemic blood pressure significantly delays proteinuria and end-stage kidney disease, but these are not a cure for Alport syndrome, though they are extremely helpful and have really changed the way that this disease is treated by nephrologists and the impact it has on patients' lives. So Alport syndrome is a disease of collagen, a particular type of collagen called collagen 4 that is quite specifically found in basement membranes. Other collagens are found in bone and skin, but collagen 4 is the basement membrane type 4 collagen. And patients that have Alport syndrome have a very stereotypical defect in their glomerular filtration barrier. The glomerular basement membrane becomes split, thickened, layers can be detected within that is never seen in the normal kidney glomerular basement membrane. So there's a word that pathologists used or used to use called pathonomic, meaning if they see this in a patient that has blood in the urine and proteinuria, that's likely a diagnosis for Alport syndrome. So I'm going to talk a bit about the genes and the proteins that are involved in the pathogenesis of this disease. So this disease is caused by mutations in the type 4 collagen genes alpha 3, alpha 4, and alpha 5. So there happen to be 6 collagen 4 alpha chain genes, 1 through 6. Those 3 genes make 6 different proteins. Those proteins can assemble with each other inside the cell to form 3 different what are called heterotrimers. And collagen is considered to be a coiled molecule that looks somewhat like this schematically. The alpha 3, 4, 5 chains are made by podocytes, and within the glomerulus, they're only made by podocytes, and those form the type 4 collagen in the glomerular basement membrane. The other genes, alpha 1, 1, 2, is found in the mesangial matrix, that I won't talk about at all, as well as in other basement membranes throughout our bodies. And then the alpha 5, 5, 6 trimer is made by the cell that makes that Bowman's capsule that surrounds the glomerular filter that I mentioned earlier, that I'll bring up again shortly. So again, Alport syndrome is caused by a mutation in any one of these three chains, because if any one of them is missing or defective, this obligate heterotrimer, three-part protein, can't form if one of those chains is missing. So because the alpha 5 gene is X-linked or sex-linked, it's on the X chromosome, mutations in alpha 5 cause the most prevalent form of Alport syndrome, and boys are more severely affected than girls, who are usually their mothers or their sisters or their daughters. In contrast, mutations that affect the alpha 3 and alpha 4 chains of type 4 collagen, those genes are autosomal, so both copies of the two chromosomes need to be affected. Homozygous mutations cause this rarer form of Alport syndrome that affects about 20% of Alport patients, and boys and girls are equally affected by those mutations. So it's a very interesting disease from a human genetics perspective. So in 1994 or so, I made a transgenic mouse, or a knockout mouse, that had a defect in the collagen 4 alpha 3 gene. And those mice, I was hoping to see a defect at the neuromuscular junction, but those mice had basically the mouse version of Alport syndrome. They have a thickened and split glomerular basement membrane. They have, at late stages, they have blood in their urine, they have lots of protein in their urine, and their kidneys fail. So what I hypothesized, what many people have hypothesized, is that the road to a cure for Alport syndrome is paved with fixes for these glomerular basement membrane defects. The defective composition, this basement membrane is missing the alpha 3, 4, 5 chains of collagen, and the defective structure, which I think is even obvious to my mother. So in 2012, we began asking the question, can this defective glomerular basement membrane, which might not be discovered in a child with hematuria until well after birth, can such a defective basement membrane be repaired? So we designed a very, what's that word? We have a proof of concept study where we generated transgenic mice where we could basically feed the mouse a antibiotic called doxycycline, and we would basically fix the defective collagen 4 alpha 3 gene. And so we did that when we began inducing basically the rescue of the missing gene between 0 and 3 weeks of age. The mice lived a long time, whereas their litter mates or brothers and sisters and cousins that didn't have this rescue reached kidney failure over a period between 15 and 30 weeks of age. So this basically showed that, and we knew that at 3 weeks of age, even at 0 days of age, the kidney is already functioning and making urine. So we were able to change an abnormal glomerulus to one that has the normal collagen 4 composition. So the Alport GBM can be fixed after it's already formed and functioning, and this supports developing therapies for patients using innovative approaches to either fix the COL4 gene defects, activate COL4 gene expression, or bypass mutations in COL4. So I'll briefly talk about two of the approaches that we're taking under the leadership of Kohei Omachi, who is a postdoctoral fellow in my laboratory. So what do I mean by activate COL4 gene expression? In a patient that has, say, an alpha 3 mutation, we can't activate a defective gene because that wouldn't really solve the problem. So the idea here is that we're going to try to activate expression of the alpha 6 gene in podocytes, which they don't normally ever make, and that will allow the podocyte to make the alpha 556 trimer, deposit it into the defective glomerular basement membrane, and maybe at least partially rescue the absence of this trimer. So this is good for patients or mice that have autosomal recessive Alport syndrome. These genes aren't affected by mutations that affect these two genes. And so we hypothesized that forced expression of the alpha 6 gene will lead to production of this trimer by podocytes and fix the GBM structure and function. So we're using a 21st century Nobel Prize winning technology. In 2020 for chemistry, the Nobel Prize was won for this CRISPR-Cas9 technology. Many of you know that Cas9 can be guided to any position within the genome, and it's an enzyme that cleaves DNA. It can induce mutations or induce a break that will allow a repair piece of DNA to be inserted. But we're using a different version of this, whereby we take this Cas9 protein that has been deactivated for its nuclease capability, but it can still bind to a guide RNA that will bring it to a particular site in the genome. And this dead Cas9 has been fused to transcriptional activators that will promote production of the gene where the Cas9 is brought next to by a specific guide RNA. So Kohei spent a lot of time designing guide RNAs, shown here as these little polygons, adjacent to the collagen 4 alpha 6 transcriptional start site, with the hope that these guide RNAs could bring this Cas9 activator to the promoter region and drive expression of that gene. And I don't expect you to read any of this, but you can see that some of the guide RNAs had very high activity, this particular one here. But he decided to combine this with two of these others and so this is a 600-fold activation of the alpha-6 gene, whereas this is a 5,000-fold when three of them are used together. So this seems to be a good way to force expression of alpha-6 in podocytes. So we made the transgenic mice that I won't go into detail about and bought some from the Jackson Laboratory, which sells mice that we need. And I hope you can see, it's a little dim, but normally alpha-6 is only in that Bowman's capsule which surrounds the glomerulus, that filtration apparatus. And we already knew that, so when the guide RNA is missing, there's no abnormal expression of alpha-6, but lo and behold, when we have the guide RNA present and stained for this alpha-6 chain, it's now found within the glomerular basement membrane, as well as, for some reason we don't understand, tubular basement membranes. And so we're now in the process of trying to do this in an Alport syndrome background so that we can attempt to rescue those mice from the kidney disease that we know they will develop. My second scientific point is about bypassing existing collagen 4 mutations that stop synthesis of the needed collagen protein. Many such mutations, called nonsense mutations, cause Alport syndrome because they stop production of one of these three proteins. So the idea that coaxing the protein synthesis machinery, which is called the ribosome, to ignore premature stop codons is a potential therapy for patients with the right kind of mutation in a personalized medicine approach. So in brief, when a patient has a premature termination codon that makes only a partial protein because the ribosome is making the protein off the RNA, and then it stops before it gets to the very end, which is normally down here, the idea is that there are certain drugs that are known to tweak the ribosome to force it to put in an amino acid to make the protein continue at this premature termination codon, and then make a full-length protein that should be functional. And so Kohei, in a brute force approach, identified 49 mutations that cause Alport syndrome in the collagen 4 alpha 5 gene and put them individually into some expression vectors and tested their ability to respond to a potential read-through drug that is called G418. It's also an antibiotic. And what he found is that there were a number, so the higher the bar, the better. There were quite a few that had significant increased read-through activity, mostly those that had the UGA mutation that causes the ribosome to stop translation. So if we could get ribosomes in Alport podocytes to read through this UGA stop codon, that would allow the cell to make the protein that's needed to make the basement membrane normal. So we published our results in Cell Culture earlier this year in a journal called iScience. And I was told by a collaborator at a company called Elox Pharmaceuticals that based on those results that we published, they decided to move forward with the clinical trial to try their proprietary read-through drug called ELX-02 in patients, a small number of patients that have Alport syndrome, to see if it would be effective. So this was quite gratifying that Kohei and our research was recognized. So for the future, I received an R01 grant from the NIDDK and funding from the Alport Syndrome Foundation to continue trying these innovative approaches to treating Alport syndrome in the coming years. We'll use 21st century technologies to discover new opportunities for therapy in proof of concept preclinical studies in mouse models that will someday help patients with Alport syndrome. That's definitely our hope, which is part of the Alport Syndrome logo, Alport Syndrome Foundation logo. So that's all my science. I'd like to express some special acknowledgments to the renal division, Division of Nephrology here at WashU, especially our fearless leader, Dr. Ben Humphreys, who led us through designing a new research space and a move to a new building that was very too quickly followed by the COVID-19 ramp down, and lots of worry, at least for me. But I think we came out of it even stronger thanks to his efforts and level-headedness. Special acknowledgments to patients. Part of me always wanted to go to medical school, and I think being involved with patients that have the disease that I study and hearing their life stories and what they have to deal with has really somewhat satisfied that inclination. When I was first hired, the nephrologists in the renal division told me that they barely ever see any patients with Alport syndrome, which in a weird way I felt somewhat discouraged by that. But meeting activist patients like these two, Sharon Lagas, who co-founded and is past president of the Alport Syndrome Foundation, and Susie Gear, a patient in the UK, who founded Alport UK. Getting to know their families, living with the disease that I study has made the research so much more meaningful and rewarding, and I thank them for including me in their efforts to educate their patients, educate them and their families and other patients, as well as clinicians about Alport syndrome. ASF asked me to host a Midwest Alport syndrome patient meeting here in St. Louis in 2016, which I was thrilled to do. You can see some of them here. And also thrilling was that they got to visit and meet with people in my laboratory. Hani Suleiman, shown here, who now has his own lab in our division, explaining to these patients with a severe kidney disease why they have that disease and what we're trying to do to slow the progression or halt the progression. I also acknowledge camaraderie within my field. ASF has put together a scientific advisory research network under the directorship of Lisa Bonebrake and Andre Weinstock, both patients with Alport syndrome, and there have been numerous collaborations among my colleagues. Special acknowledgements, work-life balance. I've been in a wine club since 2002. Some of them are here. It's been wonderful. I have wonderful neighbors. Two weeks after COVID lockdown was my birthday. Maybe the saddest birthday I ever had. But they went to the effort to have an outdoor party, socially distanced, and this kind of became our whole COVID balcony happy hour parties, get-togethers, so that we all didn't go crazy inside our own units. Of course, I'd like to acknowledge my family. I'm so thankful that they're here. My father succumbed to COVID in July of 2020. He ended with basically acute kidney injury when he went onto the respirator. We're all sad that he's not here today, but we're all here today. And also thank my brother out-of-law, Keith McDonald, for travels to colder places than we do traveling to sometimes warm places. Finally, I'd like to thank my partner, Larry, of 27 years, who balances my life. He's the Ghirardelli brownies to my Mission Figs. He's the disco to my NPR. He's the GSN to my CNN. For Big Bang Theory fans, he's the Penny to my Sheldon. He's the Barbra Streisand to my, well, there really is no comparison, is there? And finally, he's the Broadway to my opera, which sounds good for the effect, but in all honesty, we both love Broadway and opera. So I thank him for all of his support and the patience required to live happily with a scientist. Actually, finally, I would not be here today without the Graduate School Admissions Committee of Washington University in the early 1980s. Here's my letter that I received from them. I'm sorry to inform you, we cannot offer you a position in our program, the DBBS. So I was rejected from grad school at Washington University. And I called the director and he said the admissions committee felt that you did not know what you were getting into. And I can honestly say they were absolutely right. But if they hadn't rejected me, I probably would not be here today because my life would have been totally different. So thank you so much for being here, for your attention. For people who came in from out of town, it's just been a wonderful experience so far.
That was a really wonderful talk. I think we have time for a couple questions. If you want. I'm sure mostly the nephrologists in the room.
Ambra Pozzi.
I wanted to ask you, when you force the expression of the alpha-6 gene, do you automatically force the expression of the alpha-5?
The alpha-5 is already made in podocytes, so there might be a little bit of extra activity, but I don't think so. These were really designed to focus on the alpha-6 chain. But it wouldn't be a bad thing if alpha-5 were forced also. Sure.
Any other questions? If there are no other questions, we're going to have the Slatopolsky and the Minor families get some pictures down here, right? Am I saying the right thing? I'm standing in for the dean who had an emergency phone call. And then we're having a reception out in the lobby. So please come out into the lobby and enjoy while they get their pictures taken quickly, and then they'll be out to join us. That was a great talk.
Thank you.
Really, really